All right. Hey, everybody. Today I'm going to do your summary review for enzymes, what you need to know for your tests, the basics, give you some resources for videos you can watch. Otherwise, you guys should look at all the PowerPoints I've uploaded, check out our YouTube channel and the playlists that I have for the biology class. And if you have any further questions after this, do your own Google research, email me with questions, and I will do my best to help you guys remember this stuff from when we learned it in the beginning of the year. So, enzymes, as you recall, are a type of macromolecule. They are proteins. So, not all proteins are enzymes, but all enzymes are proteins. Enzymes work to speed up reactions. They either fuse molecules together, that is, they build molecules in synthesis, or they break molecules apart. That's called lysis. Enzymes are like little Pac-Men, and they go and they attach to whatever substrate they need to build or by piecing together or break apart. It's called a lock and key fit model, so usually enzymes are very specific to the substrate that they bind to. So the enzyme lactose is really only going to work on the sugar lactase. Likewise, the enzyme sucrase is really only going to work on the sugar sucrose. And I'm sorry, lactose is the sugar and lactase is the enzyme. So most enzymes end in ASE. So if you see catalase, polymerase, that's the enzyme that does polymerization for DNA polymer. If it ends in ACE, it's usually an enzyme. And if it's an enzyme, then we definitely know it's a protein. And what are proteins made up of? Amino acid monomers. Just a whole bunch of review there for you guys. So you need to know what enzymes are, how they work. Enzymes work by speeding up reactions. They are catalysts, which means they speed up chemical reactions. So those chemical reactions would happen normally without the enzyme present, but the enzyme being there speeds it up by lowering the amount of energy that is needed. So they kind of work as a booster for chemical reactions. So your body, one example that we talked about in class was how your body doesn't usually break down the sugar lactose that comes from cow's milk very quickly. That's why we have the enzyme lactose that helps speed that process up. People who are lactose intolerant, they don't have a lot of the enzyme lactase, or it doesn't work properly in their bodies, so they are lactose intolerant because their body can't break that sugar down as quickly. So enzymes function to speed up chemical reactions by lowering the amount of energy that is needed for that reaction to take place. There are some graphs that you need to know for enzymes, and you can view all those different graphs on the PowerPoints that I've uploaded and in the Amoeba Sisters videos in our biology playlist for this channel. You need to understand what the graph looks like when activation energy is lowered. So that's going to have two lines, and one of them is the reaction that is sped up when the enzyme is present and lowering that activation energy. The other line above it is how the reaction takes longer when the enzyme is not present. Therefore, it has an increased activation energy. Other than that, you need to know how enzymes work. So they bind to their substrate, and then they either build molecules by putting them together or break molecules by lysing them and then creating two new molecules from the one that was broken apart. There are three graphs that you need to know. So we need to know how enzymes work at specific pHs, and that is very specific to the enzyme. Some enzymes work at low acidic pHs, like the ones in your stomach. Some enzymes work at very high basic pHs, and that is all very specific to whatever type of enzyme you're looking at. Enzyme pH graphs usually look something like this, where their optimal range, that means the most functioning that we're having, the most activity of those enzymes is happening at whatever that specific pH is. So we would start with our acidic pH is down here around 0 and then moving up to 14. This specific enzyme functions well at a very neutral pH of around 7. We also need to know how temperatures can affect enzyme activity. So when it gets too hot, an enzyme, which remember enzymes are proteins, which are coiled up chains of amino acids that have a very specific shape. When they get too hot, they can denature because the bonds that hold them together will break due to the heat. And when an enzyme denatures, it loses its shape and it will no longer function anymore. It's done. It cannot be used. If it gets too cold, an enzyme can slow down, but if it's brought back up to its optimum temperature, it will work again. Again, enzyme temperature graphs typically look very similar to those of pH temperature graphs, where an enzyme will function best at its optimum temperature. When it gets too cold, it starts to slow down and not work as fast. And then when it gets too hot, it will denature and eventually fall apart and no longer be functionable. Finally, you have to understand how concentration of either your enzyme or your substrate looks on a graph and how it works. So remember, enzymes bind to their substrate with a lock-in key fit. If you have a whole lot of enzymes, if the concentration of enzymes is high, they're going to work as fast as they can to bind to all of the available substrates. And if the substrate concentration is high, enzymes are going to bind to them as fast as they can. Once all of the enzymes are in use, or once all of the substrates have been bound to, we're going to get a graph that looks like this. So the activity of the enzyme is going to increase, or the binding of the substrate is going to increase until you reach the point where all the enzymes are filled up with substrate, or all the substrates that are there are bound to available enzymes, at which point the reaction rate will stabilize. It's not going to slow down, it's not going to stop, but it's also not going to continue to increase because all of the enzymes or substrates are saturated. So we get a graph that looks like this with a plateau. That would be what we see for concentration of enzymes, and then we would see one like this for temperature and for pH. Again, please look at the PowerPoints I have uploaded with all the notes, watch the Amoeba Sisters videos, and ask me questions directly through email if you have any more concerns regarding enzymes. Study this material, and there will be a review quiz on macromolecules and enzymes next week, and you will see this again on your final exam. All right, guys, have a good weekend.